Hi, my name is Amy Holmes. This is my Unit 3 DNA, a new way to translate DNA beyond the genetic code presentation. In the 2019 review of the article by Giorgio Bernardi in Bioessays entitled The Genomic Code, a Pervasive Encoding, Molding of Chromatin Structures and a Solution of Non-Coding DNA Mystery, Bernardi speaks about non-coding DNA sequences that do not code for an amino acid. Most non-coding DNA lie between genes on the chromosomes and have no known function, while others called introns can be found within genes and plays a role in the regulation of gene expression. Examples of those gene expressions would be how much a certain gene is turned on, where they turn off, should they turn off, how the DNA is packed into the chromosomes. When non-coding DNA was first discovered, there were three main explanations as to why they existed. One was they were pseudo-genes or junk genes with no real purpose. The second was that they were selfish genes or selfish DNA, where they were considered to be molecular parasites to their host with no known function. And this occurred, they thought the junk and the selfish occurred between 80 to 90% of the time they were found. The third, the majority, 80% of genomes took part in at least one biochemical RNA or chromatin associated event and at least one cell type. However, with the discoveries of isochores involvement in the formation of chromatin domains in spatial compartments, it was discovered that there was a correlation with isochores and their location as far as the expression of regulation in the gene. Bernardi's paper talks about non-coding DNA helping with the folding of the 3D structure of DNA evident by looking at the long strands, which consisted of about 100,000 base pairs long of DNA, and found the unique structure called the isochores, which can then be seen and be more defined by their GC content and separated into lamina associated domains or LAD that have low GC content and topographically associated domains or TADs that contain high GC or high gene content, which the topographical associated domains are associated with peak that are created in DNA, and they have open chromatin structures. Bernardi's hypothesized that these open structures help with the transcription of the gene by allowing them to be more accessible to proteins and enzymes within the nucleus. My personal opinion on this paper after reading it is without this data, we would maybe still be postulating that non-coding DNA served no purpose still in the DNA structure, when in fact it helps to make the 3D structure of the DNA and plays an important part in it. So without that evidence and that data, we would still be in an opinion that it played no part. So I think this paper was very important in that aspect, that it helped to solidify non-coding DNA plays a very, very key part in DNA structure and its role, it allows it to be more accessible, and without that 3D structure, DNA would not be what it is. So that's a huge part of what makes DNA the way it functions. That concludes my Unit 3 presentation.